where we are left initially. That is where we are starting. Okay, now welcome again. This is intercellular junction, glands, and membrane part of histology. And as we had already, we've covered that histology is mainly the study of tissue. So we've covered variety of chapters, and this one is the second last chapter where we are going to talk about intercellular junctions, glands, and membrane. Actually, this is the most and very chief area that triggered our concept, and that is this theory is so important. Therefore, we are expected to accord maximum attention where possible. Now, let us be together. Now, first of all, I have to outline in front of you. You are able to see the outline of what you are going to adhere to when you are discussing. That is the intercellular junctions. You are going to talk about the intercellular junctions, and that is what you are able to see from your hand. We have tight junctions, desmosomes, the gap junction as well. So we'll go again and check on the glands, right? And you see what glands entail, and finally we'll do the membranes. Now here, the main objectives that are going to guide us, first of all, after this section, you need to be in position to at least describe the junction that all cells and tissues together, describe or define different types of glands. You're also expected to describe the typical anatomy of a gland, how it looks like in terms of structure. And you're expected as well to name and compare different modes of glandular secretion. So you need to know this gland is responsible for this secretion and why, and what does this gland does, what name that is it given, and so on and so on. So the most important segment here is that let us discuss it. First and foremost, let's start. What do you understand by intracellular junction? Anyone can give an answer. What do you understand by intercellular junction? I hope I'm audible enough. Nicola. Okay. Yeah. Yeah, that is quite correct and also very, very important to note. Any other person with additional complement, what you can add on top? Maybe subtraction, maybe we want to correct. Yeah, that is that. So what about tight junction as example of intercellular junctions? Can someone tell us anything about tight junction, desmosome, gap junction, how they relate and how they differentiate from one another? Okay, let me do this. Now, first of all, when you talk about intercellular, that is not intracellular. Intracellular is something to do within the cell, but intercellular is between a cell. So actually, cells are not continuous because they are numerous, they are millions, they are trillions, they are billions. You can find even in a single tissue or organ, we have trillions of cells, and it's not just a large cell. So within each of the cells, they are adjacent, they are neighboring one another. So they have a space between. That is what we call the intercellular space. So within the intercellular space, there are junctions, and these junctions are very important because they consequently they link the cells. They communicate. The cells communicate through the intercellular junction. So according to what you are able to see from your end, we have three types of intercellular junctions, and these are very key things. Now, here is a case. We have tight junction, desmosome, gap junction, as you can see them from your hand. Now, most cells, with the exception of blood and metastatic cancer cells, the remaining cells, apart from the blood cells and the metastatic cancer cells, must be anchored to each other. And apart from being anchored to each other, nobody, even yourself, you cannot survive alone, right? And that is even the cells, they cannot survive alone because they have to communicate. And so they are anchored to one another and again anchored to a matrix. So if they are to grow and divide normally, that is what is very important. They have to relent and rely on the cells and also on the matrix. So the connection between one cell and another is what we call intercellular junction, that junction, right? So these attachments enable the cells to resist stress and communicate with each other. So when, for example, when you get a trauma and then you fall, then the cells within the injured or targeted areas, they are supposed to ulcerate. But in this case, they will try to maybe compress one or find a way to absorb the stress. And also they will communicate with each other and aid on the response through inflammatory processes, exchange of substances, and through the various mechanisms. So without them, like for instance, cardiac muscle cells will be pulled apart when they contract and they will tear, right? And even every swallow of food will scrape away the lining of your esophagus when you're eating without that communication, that linking of intercellular junctions, right? So the principal types of intercellular junctions are what you're able to see from your end. We have intercellular junction, tight junctions, desmos, and gap junction, or what we call communicating. So let's start with the tight junction. Anybody with anything about the tight junction? If you even if you don't have any theoretical answer, you can relate it with anything possible, so that you can give us that view. So any other opinion? Anybody with idea on what is tight junctions? Kariuki Olivia, welcome. Tell us anything. Okay, somebody somewhere try. Yeah, they're mainly found in epithelial tissues. That is good. That's a nice trial, actually. Any other person, please? Yes. Yeah, I can get your point because they are tight. They regulate. So you see, regulation composed of resisting others, restraining others, and allowing others. Okay, that is bringing another idea. Okay, so up to there, let's see on what are tight junction. First of all, what I want to know from the word tight junction, these are completely encircled and epithelial cells. Actually, it gives out entire captivation of the epithelial cells. And you'll see it when I show the entire image of all of these tight junction, desmosop, and gap junction. So next to the apex of the epithelium, you'll find something encircling and joins it tightly to the neighboring cells. Like, for instance, when you're in high school, you can remember when you're talking about, yeah, we had two kind of tissue which were helping transporting water and translocating food. The phloem, yeah, the phloem. There was somewhere there written desmosomes, and you could see it links to the sieve tube and other adjacent parenchymal cells. So that desmosome was one of those intercellular junction, but it wasn't tight junction. But for the tight junction, they are very tight, right? Like something like a plastic harness on a six-pack of soda cans. You see how they are always tightened. So the proteins in the membranes of two adjacent cells, they are protected from zipper-like, something like zip. I hope you know you've seen zip of long trousers, yeah. So there's a zip-like pattern of complementary grooves and ridges, so which are interlocking. So this will seal off the intercellular space and make for some substances to pass between the cells. So in the stomach, for instance, and intestine, yeah, if you have a keen move into your morgue where you're practicing or in the clinical session, I do like actually going to the morgue most often. Why? Because that is where you can see directly the tissue and what it entails. So let me just allow these guys who are on the waiting bed so that they don't complain. Let me bring them in. Okay. So that is that. So I was saying when you go to the morgue, huh? At most, when you check on the stomach and the intestine, you realize that tight junction prevent most of the mechanism that we know, like digestive juice from seeping between the epithelial cells, digesting the underlying connective tissue, that is. So they are very protective, especially in the stomach and the intestine where they are mostly found. They also help to prevent things to do with intestinal bacteria from invading the tissue. Remember when somebody is on a Clostridium or on any other, even the dysentery form of even amoebiasis, but no, some are very little than even the tight junction. Like when you say amoebiasis, it has a portion of it which is invasive and non-invasive. So definitely it will tear apart the intestine. But what I want you to know is that the main function of the tight junction, like in stomach, is to prevent digestive juices from seeping between the epithelial cells and digesting the underlying connective tissue. It also helps in preventing intestinal bacteria from invading the tissues which are surrounding the intestine. Tuko pamoja? And they ensure that most digested nutrients pass through the epithelial cells and not between them. Because if at all, the spaces of the tight junction were quite big, instead of the digestive nutrient or digested nutrients to go inside the correct route, they could have been sipped all through the tight junction without, consequential procedure that they're supposed to be following. It's all about tight junction. Let us go to desmosomes. Anybody with an idea what is desmosome? Before we see the entire image, anybody with the idea what are desmosome? Yeah, it gives a mechanical frame to a tissue. That is what you say, right? Yeah, some sort of. Okay, actually, they're just somewhat similar. Any other person with an idea again? You are good. That is another good opinion. Any other person with another one? Uh-huh. Yes, we are getting you. Proceed, please. Yeah, so Ruth Mwang is telling us that it contains two adjacent filament. I mean, is it using the filament? Is it practically filament? Okay. Yeah, they're quite the same, right? But you can realize that a desmosome which is in a given organ, like for example, which one can I use? Just let's say a surface of a typical cell will be different from another cell. So some will use the filament, intermediary filament, like and you remember the cell has got when she was talking about the filament, you could have configured something like cytoskeleton, the structure, the cytoskeleton structure that give the cell integrity. When you are launching the cell, you could see that whole component of a cell. So here, let's go. You see, if a tight junction is like a zipper, now a desmosome, or what we call, the correct pronunciation is supposed to be desmosomes, yeah, is more like the snap on the pair of a jeans. I don't know if you know a snap on a pair of jeans, a patch that holds cells together like and enables a tissue to resist mechanical stress. So what they have said is correct. Like they are giving the tissue some of a cajoling mechanical power, but it does not totally encircle a cell. They just give a portion of place that energy. So I'll give you a redirectory on where you can access entire image of such desmosome. So at later on when you're free, you just text me. Sometimes I do forget. Yeah. So desmosomes are common in the epidermis, right? Cardiac muscles and the cervix of the uterus. We have epidermis. If you are writing, you can put down epidermis, cardiac muscles, and cervix of the uterus. And then the neighboring cells, when we are talking about desmosomes, right? The neighboring cells are separated by the small gap, which is spanned by a fine mesh of glycoprotein filament. And if you're talking about glycoprotein filaments, I hope you know what we mean. So these filaments, the way she was saying, they will terminate in a thickened protein plaque at the surface of the cell, and now not only in one cell but each of them. Utapata ziko kila mahali. So then on the cytoplasmic side of that cell, of each plaque now, we'll find what she referred to as the intermediate filament now. That is directly linked to the cytoskeleton. So it approaches from the cytoskeleton approach and penetrate the plaque like. So it gives out a passage like. So it returns shorter distance back into the cytoplasm, giving a passage like. So that is what we refer to as the desmosome. So each cells will contribute half of the complete desmosome. So not that one is entirely having a whole. No, you will find that the cells are too much close in a way that each of them have attributed. Zote ziko na mapacha ambayo ziko na shimo. So zikishikana unapata ni kama is a complete gap. That is why we call it the snap on a pair of jeans. So I've said where you can find these things are in the cardiac muscles, cervix, and uterus. And the right way how you can see how it works in literal aspect by viewing a video Go to YouTube. I know there are good people there and also there are bad people. So just research closely, checking on how desmosomes work or the physiology behind desmosomes in a cell. If you type that, that. So hope we are good. Are we good up there? Can you proceed? Am I audible? Okay, sir. So I wanted to think about something to do with why will desmosome not be suitable as the only intercellular junction in the epithelium of a stomach? What do you think? Can we think about it right now? Why is it that the stomach behind the desmos still have tight junction? Or let me just leave that on assignment. You will think later. Okay, so let's go to gap junction. Who can say anything about gap junctions? So on that, Mhm. Yeah, when you look at gap junction, you just find it's very funny why they brought gap junction and tight junction together. But they are quite opposite of one another, but they are also important in the physiology. That is why you find, beside the fact that the tight junction are so responsible for tightening the spaces, gap junction will permit, but considerately. It is molecularly formed and hypothesized for its function. So, what I'm trying to say, or what I need you to understand is that gap junction is formed by a ring like what we call the connection or connection. So it consists of 6 transmembrane proteins. And when we talk about transmembrane proteins, I hope unafikiria ile kitu inapita direct from the, in the cell membrane we have the lower part, the peripheral and the inside. Oh, so it passes across it's a transmembrane. So I'm saying it has got 6 transmembrane proteins surrounding a water-filled canal. And therefore ions, glucose, and amino acid and other small solute can pass freely directly from the cytoplasm of one cell into the next through these channels. So let's go back. When we are in the embro, I don't know if you know what I'm saying, but embro, that is what embro, yeah, from zygote to embro, not embro, right? So in the embro, the nutrients pass from the cell to one cell through gap junction until the circulatory system forms. Because division of a cell or mitosis and the mitosis process is okay. I don't know if I'm on, but suggest am I audible? Just in a few, I'll be back. Hello, am I now audible? Hello, I hope you're able to get what I'm saying, right? My internet went, but I hope it is back. I hope I'm audible, am I? It was off and it is now on. Hello, are you able to hear what I'm saying? Okay, now let's proceed. If you're good now, let's proceed. Initially, we were talking about gap junctions. I was talking about the 6 transmembrane, which was passing over through, and they were like proteinous in nature. They could allow things to do with the glucose, amino acid, which are important for the cell. And again, I devolved and say I was talking about embryo. Giving an example, when the circulatory system is not yet developed, there are a lot of questions, like for the survival of the cells, because each of cells which are divided, no matter how they are not yet complete and they don't have any sense, they have to be provided with oxygen and nutrients, and they have to communicate. Not talking about circulatory being incomplete, it means even the blood vessel cannot link the cells. So what normally is important here is that the gap junction normally take the role. So the gap junctions, they're the ones which are now responsible during that stage of the embryo. So the nutrients will pass from one cell to another through the gap junction until the circulatory system forms and takes over the role of nutrition or nutrient distribution. So gap junctions are found in the intercalated disc of the cardiac muscles. Are we together? And between the cells of some smooth muscles. Again. So I've said in the cardiac muscles and also in the smooth muscles. And then the flow of ions through these junctions allows electrical excitation to pass directly from cell to cell. So the cells contract in a near unison. So like when these gap junctions are playing a role in the intercalated disc, in the cardiac muscles, they enable that unison, which I was telling you, the contraction and spread of the action potential should be uniform and at once. So contraction in the muscles are apprehended by the gap junction, which enable them to contract at once. Now, before I proceed to that is glands, I'll first show you the image of all these the tight junction, desmosomes, and the gap, and then you see how they look like. And I'm sure that from your hand you're able to see what I'm talking about. That is what I'm talking about. Are you able to see the image? So there you'll see we have tight junction, plasma membrane, membrane protein, intercellular space. That is in the tight junction. And then we'll have desmosomes. And you see how desmosomes look like, those plaques, the plaques which I told you, yeah? The plaques will now give us, each of the cell will play a role because, each of the cell will have a plaque and the other plaque, and therefore when they, so a complete desmosomes must be contributed by two cells. So between fun, we have intermediate filament, the intercellular space, and all those things. Then the gap junction, there's a pore, which pass between one cell to another. So they are communicating. Those are the tight junction, desmosome, and gap junction. So if they come in anatomy MCQ, you will know how to answer them accordingly. I remember during our time they came. So anyway, let's proceed. Let's proceed to the next. And before we go, there's a condition I wanted to talk. Every day if you're discussing anatomy or physiology, you have to relate a concept with a medical condition. So there's a clinical application of what we call the pemphigus vulgaris, and you can see it from your hand. The clinical pemphigus vulgaris. This is an autoimmune disease. Now, what do you understand by the word autoimmune? Anybody anywhere? Autoimmune disease. What can you say about autoimmune disease? Medical students are relevant. You guys, any other person, immune system imefanya nini, my dear? Immune system, it's supposed to be a normal, actually an important attribute. Listen. Niko class a minute. So, tell me. Yeah, that is now at least. Yeah, you can't just see caused by immune. That is not at least. When we have antigenic properties, which are supposed to be elicited by the microbes. At most, each bacteria has got its antigenic property. That is why even antibody only attack a specific uh, bacteria. Just a minute. Jeremy. Yeah, yeah, yeah. So, I hope we are together still. Yeah, I received a call. Okay, so let's proceed. Now, who can tell us in details? I was talking about an autoimmune disease. So, like, let's say bacteria. Let's say we have Neisseria gonorrhoeae or mirabilis or maybe chlamydia, or a simple term, the one which you're commonly used to. Let's talk about a syphilis. And The most key thing is that each of these things they will follow the clinical, history of disease. That is from the entry, the latency, right, as we proceed henceforth. So up to the clinical stage, we also still have asymptomatic and symptomatic. Now, the antigenic property of which microbes will tell the immunity of the body to attack specific, in a specific way, in a specificity manner. So, like if your body mistake your own cell to an microbial antigen, it will attack itself. So that is what we refer to as autoimmune diseases. So most of autoimmune are congenital, some are being you're born with, but some are acquired externally. I hope you are good. I don't know who is having his microphone on. Uh, I hope we are good, right? So that is that. So let us talk about the pemphigus vulgaris. Here is another autoimmune disease. So this immune disease that the system normally the immune system is supposed to produce defensive antibodies against the antigen. So that is selective attack foreign substances. That is what I refer to as the antigens. And so they will leave the normal tissues of our bodies alone without causing harm. But in a family of disorders called autoimmune diseases, what you can see there, the antibody failed to distinguish our own cells and tissues from foreign ones. So such misguided antibodies they are called autoantibodies. Thus, launch destructive attacks on our own bodies, and that is what is here. Now, let's talk about this one. The pemphigus vulgaris is a disease or a disorder in which antibodies. Can you please mute yourself? Just a minute. I mean, Nelson, somebody. Nelson Masiga, don't unmute yourself. I've mute you, so keep yourself like that until you are supposed to answer any question. So that is that. So I was talking about the pemphigus vulgaris, and I've said the autoantibodies attack the proteins of the desmosomes in the skin. So in this case, the mucous membrane inaanza kuharibika. So it breaks down the attachments between the epithelial cells and causes widespread things to do with the blistering of the skins, yeah, oral mucosa, loss of tissue fluid, and sometimes death. So the condition can be controlled with the drug that suppress the immune system, but such drugs will reduce the patient's immune defenses against other diseases. So we know of these drugs, yeah, which can control. They will suppress the immune system, and when we suppress our immune system, what normally comes in is that now opportunistic are given a priority, so they'll play their maximum role. So that is that. So we are going to glands. Remember, the entire topic was to talk about the junctions and the glands. So we are going to glands, and then finally we are going to talk about the membranes, and then we are done with this one. Now, we have talked about the intercellular junctions. Now we are in glands. We have the following words which supposed you can see from your hand, and I want somebody to distinguish between endocrine and exocrine glands. And again, you talk about each of them. You talk about the endocrine and exocrine gland. Then, Nicole, are you there? Yeah, you remember the question you posted in my inbox, eh? Yeah, it is just next year I want to tell you about it. No, that is why I had to revise the entire thing. So, may somebody tell me the difference between endocrine and exocrine glands? Wow, that is so wonderful. Yeah, another person, at least two more people. Yeah, yeah, yeah. Endocrine. directly, na hizi zingine ni glands inatema. So endocrine, bloodstream, direct. Okay. Another person, the last person, please. That's still the same thing, but now a different angle. No, more than one idea gives you a permanent understanding. One person gives you just that, admonition of the entire thing, but now from more than one person, you get a family of it. But tell me, another person, the last person, please. Okay. What I want you to know is simple. A gland is always a cell or organ that secretes substances. That is from entirely biology class. You've been knowing this one. So it can be producing this thing to be used where it is being produced, or it can be used in any other place in the body. So because it's something that secretes substance for use elsewhere in the body or release them for elimination from the body, you can also release them so that they can be eliminated, excreted off from the body. So the gland product may be something synthesized by the gland cells or something removed from the tissue and modified by the gland. I wanted to get this point. It doesn't mean that gland is every day going to produce for us something which is consumed by our body, but at times a gland is like just a secondary attribute to a functional tissue. It helps the tissue to secrete, modify the waste product of the tissue that enable it to be easily eliminated. I hope that point is at all. So glands, like for example, the ones secrete, like such as digestive enzymes, they are for consumption. But such as the ones that now make the urine to become urine, that is now for excretion. Glands are composed they have what we call epithelial tissue, predominantly by epithelial tissue. That is what composes them. Now let's talk about endocrine and exocrine gland a bit. That is what I want to tell. The overview I've already said. So let us now talk about the endocrine and exocrine glands. Now, glands are broadly classified. What I say broadly, I mean just like they have different classification, diversified classification. So either endocrine as exocrine, that is one, that is the main one, endocrine or exocrine. They originate as investigation of a surface epithelium, these names. So in exocrine gland, let's start with exocrine. They usually maintain their contact with the surface by a way of a duct. A duct. So I've said they will keep or maintain their contact with the surface by a way of a duct, an epithelial tube that will now convey their secretion to the surface, whether epithelium, externally or internally. So the secretion may be released to the body surface, as in the case of the sweat glands and mammary glands and the tear glands, but more often it is released into the cavity, that is in the lumen of another organ, such as the mouth, that is salivary gland, intestine, yeah. Now that is about exocrine. I've said they always keep their contact by the use of duct. Now let's talk about endocrine. Endocrine glands, they lose their contact with the surface. They don't maintain, they lose their contact with the surface and have no ducts at all. They do, however, have a high density of blood capillaries and secrete their product directly into the blood, the way you guys said. And the secretion of the endocrine glands are like hormones. Generally, they are called hormones. So they function as chemical messengers to stimulate cells elsewhere in the body, right? And the endocrine glands, they are the subject of what we're going to see later when we are going to reach. There's another chapter where I'm going to just talk about the hormonal system. So the exocrine and endocrine distinction is not always clear. That is why even I had started with asking a question. Like the liver is an exocrine gland that secretes one of its products, bile, that is bile, through a system of duct. But again, it secretes hormones like albumin, right, and other products directly into the bloodstream. So it's both. And that is what I am telling you. Again, several glands have both exocrine and endocrine components, such as pancreas. I know you know its endocrinal effect. And by the way, you can tell me both the endocrinal and exocrinal function. You have to say this is an endocrine function of pancreas and this is the exocrine of it. Are we together? Is somebody hearing me or I'm talking alone? Hello? Wow, that is good. That is good. But will you please tell me the hormone and the enzyme? Somebody to help. No, okay, okay. You're right. You're right, karaoke Olivia. Somebody else to help karaoke giving out specific secretion from the pancreas, both in endocrinal, notion, and exocrinal. Yeah, the pancreatic juice has got some many elements. You need to know them, right? Including the salts. That is that. You go and check on that at home. Now, very good. Let's proceed. Yeah, yeah. So I was talking about the some organs being both of these. So that is that. So even though most of these organs are not usually thought of as glands, for example, the brain, you see, brain can also secrete. So they nearly all the visceral have at least some cells that secrete hormones, and they will not mostly be called an organ. Like brain has got its own secretion. Heart has a secretion. Nani anaweza niambia secretion ya heart? It has got a very funny name, and it is used as a confirmatory test of some conditions. Okay, then somebody who will remind us because I also remember half of it. Okay, that is medical students arena. It's down. Okay, let's proceed. Higher. Let us talk about unicellular glands. They are glands with now one cell. And now the exocrine cells, the exocrine cells, I mean, they are found in an epithelium that is predominantly non-secretory. For example, the respiratory tract. It is lined mainly by ciliated cells, yeah, but it also has this liberal scattering of non-ciliated mucus secreting goblet cells. So those are what we call unicellular glands. Away from the endocrine glands, now let's talk about exocrine glands. Exocrine glands peke yao. And I hope we are together. Are we together? Okay. Exocrine gland structure. And I'm going to show it in an image, a sequence of image. So itself, we are going to see what you in front of you there is exocrine gland structure, and that is where we are. Now, exocrine gland structure is what we're going to start by. So we have multicellular exocrine gland, a structural arrangement found in such organs as the mammary gland. You find a lot of cellular within alveolus and so on and so forth. So that is what we call the multicellular exocrine gland. So yeah, like in pancreas also, it has got all this, the acina, the blah blah blah, the beta, so on and so forth. So most glands are enclosed in what we call the capsule. Hata ukisoma anatomy yako utapata even the mammary glands they have capsule. And the capsule often gives off some extension which we call septa or the trabecula. The septa you ever know, like the cardiac septum, will divide. So they normally divide the interior of the gland, yeah, into compartment called lobes. Now I'm talking specifically about mammary gland. You find the septa divided into several lobes, which are visible to the naked eye. And then finer connective tissue septa may further divide or subdivide it into other lobes. And then we call it lobules. Now cannot be visible where you are utatumia microscope. So they are microscopic, yeah. And then blood vessels will also be in structure. So apart from the septa, the capsule, now I'm talking about the exocrine structure. We have capsule, we have the septa, the tubercles, we have the lobules, we have the lobes. Yeah. Apart from there, blood vessels are also very necessary. So the blood vessels, nerves, and the glands on duct generally travel through the septa. So you need to know the component of the structure and the connective tissue, which now support the exocrine gland to the adjacent framework. So like a connective tissue framework of the gland, which we now refer to as the stroma. Now, if somebody asks you stroma, you talk about that. Now, and stroma can mean many things in medicine, so you have to be specific. So the connective tissue of the stroma support and organize the glandular tissue within the exocrine glands. And then the cells that perform that task of the synthesis and secretion are collectively called the parenchyma. So when I talk about parenchyma, these are kind of tissue that takes, that perform the task ya kuproduce, like synthesizing and secretion, performing the tasks of synthesizing and secretion are collectively called the parenchyma. So when you talk about this, we can have them group either into simple cuboidal or simple columnar epithelium, depending with how the parenchyma look like. So when I'm talking about exoglide, again, you still need to know that we have other further classification. They can be classified as either simple if they have a single unbranched duct, that is simple exocrine gland, either branch, or compound exocrine glands if they have a branched duct. But if the duct and secretory portion of for example, I don't know which example can I use, but you just get it what I'm trying to say. We have simple and compound exocrine gland, and the duct and secretory portion, if at all they are of uniform diameter, the gland is now going to be called tubular. That is why you feel some people say this is a secretory acina, this is secretory tubular, right? So I'm saying if the duct and secretory portion are of uniform diameter, the gland is called tubular. Now, if the secretory cells form a dilated sac, the gland is called acina, and the sac is called acinus. So we have the gland being acina, and the sac is now acinus. A-C-I-N-U-S. And then acina is A-C-I-N-A-R, the tubular, you know. You can call acina, acinus, or alveolus, still accepted. Acina and alveolus zinafanana, don't say there are three types, right? That is exocrine gland. So a gland with secretory cells in both the tubular and acina portion is called tubular acina gland. Meaning iko na zote, right? Are we together up to there? I should repeat. No, I'm also, there's some complex part of medicine that requires a lot of explanation. Are we together up to there? Or who can repeat? Who can say what elewa? According to what I said, please. Asante. Nani ana? Hmm? Mhm. Ni sawa. Sasa nataka kujaribu kuisema. Naona tumeongea kuhusu structure ya exocrine glands, tumeongea kuhusu capsule na itunza septa inaigawanya, then lobule ndio inapatikana. Tukishaigawanya na septa, then inaweza gawanywa zaidi ikuwe lobules. Then tukatoka hapo tukasema hiyo yenyewe haiwezi kaa peke yake hadi itafutiwe connective tissue ambayo itaisupport, na hiyo connective tissue tumeita stroma. Tukasema hii stroma ina support na ina organize hiyo glandular tissue yote. Alafu kuna cell yoyote kwa mwili yako, hata si kwa gland peke yake, ambayo ina perform hiyo kazi ya kusynthesize hizo mambo ya secretion. Zote zikiwa pamoja tunaziita parenchyma. So unaweza sema mammary parenchyma. So tunaweza kuwa na simple cuboidal parenchyma or columnar epithelium parenchyma. And then tukakuja tukasema tunaweza tena classify exocrine gland mara mbili, simple ama compound. Kama ni simple, ziko na unbranched duct, mean duct yao ni direct pale zinafaa zitoe vitu. Kama ni compound, iko na too much branch, branch. Kama you can see, they have, you can go and check types of glands around, you see example of the one with compound and simple. Yeah. And then nikasema kama hiyo duct and secretory, if the duct and secretory portion are of uniform diameter, to mean zinatoshana, yani uoni pali. Okay, hii ni mkono yako, umechukua pipe ukaweka maji huku mwisho, inaenda kumwagika nje. Pale nje kama inatoshana na hiyo pipe yote, to mean the duct na pali maji inatoka nao zinatoshana. That is what I'm trying to say. If the duct and the secretory portion where this nini inatoka nayo, if the diameter is the same, if they have uniform diameter, the gland is called tubular. Yani inakaa tu tube yote, hakuna kitu imefura juu yake. But ukiona secretory cells hapo mbele ya hiyo duct, the secretory cells zimedilate zikatengeneza sac, the gland is called acina, and the sac is called acinus or alveolus. So unaweza kumbuka, tuko pamoja hadi hapo? Tuko poa? Just a minute, I check somebody is knocking on the door. We have another thing which I was talking called the tabula acina. Tabula acina to mean kuna pati ya hizi maduct ambaye ziko kama tubule na kuna zile zimevura pale secretory, so ziko kama acina. So a gland with the secretory cells in both the tubular and the acina portion is called tubuloacina gland. End of quote. So let's go to the other thing which you can see from your hand, the types of secretions. And the types of secretion, here you need to know we have serous gland, mucus gland, mixed glands, and cytogenic glands. Though I never included them in here, but I think you can write them somewhere. We have serous gland, mucus glands, mixed glands, and cytogenic glands. So let's talk about the type of secretion in details. You need to know glands can be classified, or they are classified not only by the structure, what we were talking about in previous time, like hii inatumia duct na hii inatumia bloodstream, ama hii iko na acina na hii iko na tubular, but they can also be classified by the nature of their secretions. So like when I talk about serous glands, they produce relatively thin and watery fluids such as perspiration, right? That's the example of serous glands, such as milk, right? Tears and digestive juices. So I've said serous glands inatoa vitu ziko thin. So they have milk, tears, digestive, blah, blah. Then mucus gland. Kama ile, okay, I want to ask a very, very, very simple question. Where do we find mucus glands? Hello? Thank you so much. You're very bright. You're present. Eh? Mucus gland, they are found in the tongue and the roof of the mouth, among other places where we can find them. Those are the main ones, like which we are not supposed to forget. Now, they secrete glycoprotein called mucin, most of the mucus glands, mucin. So, and after it is secreted, the mucin can be absorbed. So it absorbs water. Mucin itself, it absorbs water and forms the sticky product which we call mucus. So mucin gets water, then form mucus. So you need to know that mucus, the secretion, is spelled differently from mucus, the gland. Mucus, the gland ni mucous, then mucus, the secretion ni mucus. And the adjective form of the word is what is used. Now the adjective of mucus is what is mucus gland. Now the other gland or type of secretion is mixed glands. And in mixed glands, such as the two pairs of salivary glands in the chin, in your chin, we have two pairs of salivary glands. They are mixed glands. They contain both serous and mucus cells. So when I talk about mixed glands, they have both serous and mucus cells, and therefore they produce a mixture of the two types of secretion. The second types of secretion or a gland is what we call genic gland, cytogenic glands. The cytogenic glands, they will release all cells. So zinatengeneza cells zinatema up. So the only example of these are the testes and ovary. Those are the example of cytogenic glands. So they will produce sperms and egg cells. Now, we are where and our friend Nicole had her issue. Nicole, are you there? So we are in method of secretion, and that is where now we'll talk about the issue. Now, I don't know if anybody can tell us anything about method of secretion before I say anything. Yes. Thank you. That's what I needed. So we have methods of secretions. So they are classified as either merocrine, holocrine, and we have acrine, or you can call merocrine or acrine. So depending with the produces their secretion, how they produces their secretion, right? And the hypocrine. So I've said holocrine, merocrine is the same. I want you to get me this. Glands are classified either as merocrine or holocrine, depending on how they produce their And merocrine glands, we can also call them eccrine. Eccrine ni tu hiyo mero unatoa alafu unaweka hapo mbele e. So merocrine can also be called eccrine glands. Now, I don't want to go fast because if I go fast. So the merocrine ziko na vesicles. And these vesicles zita release their secretion by exocytosis. And I hope you know exocytosis. And you'll see. And if I can say from the layman attribute, exocytosis is where something which is contained by the cell is expelled out from the cell by the cell itself. So outside of the cell. So just like a vesicle being formed, then moves into membrane, attached to the cell membrane, then forms and secretion portal or pinecytol, then it is inatolewa hinje. So you'll see that one when you reach in the, and I think you have already done this bulk transport in physiology and anatomy. So these include the tear gland. That one is, I'm talking about merocrine or eccrine. They include the tear gland, the pancreas, the gastric glands, and many others. On the other side, we have holocrine glands. In the holocrine glands, cells will now accumulate. So after they accumulate a product, and then the entire cells will disintegrate. So like when we want to remove a waste product, like a cell itself forms in form of that waste product, then it pile up. After it has accumulated to some level, the entire of it disintegrate. Unlike the other one, which was releasing by exocytosis, this one, they disintegrate. So the secretion is a mixture of the cell fragment and the substance the cell had synthesized prior before it disintegrates. It's just like a sacrificial excretion. I am a cell, I know there are some toxin. I will accumulate them inside me for a duration, then later on I burst and release myself together with the entire content. That is what I mean. Now, for example, the oil producing glands of the scalp they are example of holocrine secretion. So the holocrine secretion, normally they tend to be thicker than merocrine secretion. That is why most of the merocrine secretion are what we were talking about. They are somewhat serous in nature. I hope you are good up to there because I want to show you the images before I go into apocrine, but I can just do everything, then I show you the entire images once and for all. Now, apart from holocrine, merocrine or eccrine, we have apocrine. So some glands such as the axillary, and I know where you know the axillary region in your armpit, the sweat glands and the mammary glands, they are named apocrine glands. Why? We'll get to know. It is from a former belief that the secretion was composed of beads of apical cytoplasm that broke away from the cell surface. So that is why they gave it the name apocrine glands. So when they did a closer study, it showed that this is very untrue. So the glands are primarily merocrine, zinakaa tu kama merocrine in their mode of secretion. So apocrine and merocrine, they're not that far. But because people assume that they were, because of the scent that they were coming with there, so that people think that, oh, this is like just but apical cytoplasm breaking and coming along. Yeah. So these glands are nevertheless different from the merocrine glands in the function and histological appearance, how they look like and what they do. And now they are still referred to as apocrine glands, even though their mode of secretion is not that unique. That is all about glands. That is all about glands. And in the next session, or we are supposed to talk about membrane. I think this is the longest class today. I think, and it's over. So before we go to the membrane, which is the last bit of this one, I want you to check on that image. I hope you are able to see what I'm showing you. That was the first image, and as you can see, we have lobules. That was an image showing you the component of the gland, the structure, right? Yeah, the image that tells you what we were discussing initially, exocrine gland structure. You're able to see the parenchyma, the lobule, the septa, the stroma itself, and then you can see the duct and the acinas. Unaona kama hiyo duct ni tubule, then acina zimenona, so that is an acina, right? And then the other one, I told you, you can also call it tubular. And if it has got both from different, say it is tubular acina. You can see what I was talking about. Now another image is down. Let me show you another image. That is another image. You can see we have now different variety of glands. So this one here, they're just like an expression of type of exocrine glands. The color, which is we have one for duct and secretory portion. You can see the first one and its name simple, the coiled tubular, and the other one we have compound acina, compound tubular acina, you see. And then the lower picture A, there is a different notion, tells you how secretion is done. A and B, those are mode of exocrine secretion, how they are releasing the hormone. Then away from there, if you are still together, I want to know, are we together? Am I audible? Ama internet yangu imeshapotea tena? Okay, that is that. So let's go to the next. That is the last point. We have membranes. Now we are going to talk about membranes. Quite a very, very big. But are you able to read what is on your screen? All of them, eh? Okay. Now, I hope Nicole is well conversant with the terms, how classification of the glands and their secretion is when you talk about merocrine and endocrine and blah blah blah. So let's proceed. When you use an atlas of anatomy, it's the best thing you can use to evaluate yourself and confirm whether you really know and you can distinguish this tissue for you to meet your objective of the course. Now, mostly in an atlas, like we have type A atlas, atlas A, atlas, you have classification of atlas, you'll check on that. So the major cavities of the body is normally described in atlas A. And then some of the membranes that line them and cover their viscera is what we're going to. So when you talk about membrane, that is what it entails. Now, I wanted to consider some histological aspect of the major body membranes because you've been doing them, and in the first semester, in first year, you know they normally give you anatomy, which you draw some of the structure like the structure of the stomach anatomy. And so the same applies here. So the great thing here, or what I needed to know, is that the largest membrane of the body is the cutaneous membrane. Or more simply, you can call it the skin, cutaneous membrane or the skin. And it consists of a stratified squamous epithelium. And it is resting on a layer of connective tissue, which we call the dermis in the other lesson. So unlike the other membrane to be considered, it is relatively dry. And it retards dehydration of the body and provides what I told you in the other class, that it is inhospitable environment for the growth of infectious organisms. And again, I want you to understand that there are two principal kinds of internal membranes. So I've talked about the largest membrane being cutaneous membrane. Now I'm talking about two principal kinds of internal membranes. And here we have mucous and serous membrane. There are two types of membrane, either mucous membrane and serous membrane. Should I ask any question about mucous and serous membrane? Anybody who can say anything about the two? Okay, let's proceed. Okay, tell me, somebody was to speak. Hello? Can you say what you have to say? Hmm. That's what I think. Okay, it's good to know. Yeah, that is informative. You know, at a point, sometimes these things we just know, but how to put in the question. Now let's proceed and check on that. So we have internal membranes. Yeah, two principal ones are the mucous and serous membrane. So mucous membrane, we are going to start with it, or the mucosa. So when I say mucus, you can also refer to what you all know, mucosa. So it lines the passages or the passageways that open to the exterior environment. Yani zile zinafungua ndani iko nje, yani exterior environment, it relays what is inside to outside, so it exposes elements out. So it's just like aligning the exterior environment, the passageways for the exterior environment. For example, the digestive, the respiratory, the urinary, right? Yeah, I hope we are good, and reproductive tracts. And also you can talk about other tracts that you know that are they are lining or they have passageway exteriorly. So a mucus consists of 2 to 3 layers. One, an epithelium. Second one, an areolar connective tissue which we call lamina propria. See what I've written for you there, lamina propria. And sometimes after lamina propria, you'll find a layer of smooth muscle called the I don't know what you call it, muscularis, yeah? The muscularis mucosa. And then, so the mucous membrane, beside having all this structure, right, they have absorptive, secretory, and protective function. Every day, if you're told to answer any, the epithelium of the stomach, what is its function? You can say secretion, absorption, and protection. So they are often covered with mucus secreted by goblet cells. That is still on the one which is called mucous membrane. We are still in mucosa. I've said they are covered with the goblet secreting mucus. So the mucus secreted by the goblet cells, and then the multiple or the multicellular mucus glands. Or they can be both. They can be mucus gland together with the goblet cells. So the mucus will trap bacteria and foreign particles, which will keep them from invading the tissues and aids in their removal from the body. And then that is all that we know since we started learning. Then the epithelium of mucous membrane may also include absorptive, ciliated, and other types of cells. So don't worry about that, but it's something you need to know. That apart from having the mucus, can have cilia and some other absorptive properties like the villi and microvilli. So let's go to serous membrane or the serosa. We are done with the mucosa. So in the serosa is composed of a simple squamous epithelium resting on a thin layer of areolar connective tissue. And the serous membrane, they normally produce serous fluid the way our colleague has said. And that one will always help. Okay, it will help in arises, or it will arises from the blood and derives its name. That is why, okay, the name came to be serosa. You know, we have serum in the blood. It's that it was composed from the blood, it came from the blood. People think like that, and so the name came to be serous. So it arises from the blood and derives its name from that, and the fact that it is smaller to blood serum in composition. And then the serous membrane, which we are talking about, the serosa. So serous membrane, it will line the inside of some body cavities and form smooth outer surface. Yeah, so when we have a smooth outer surface of some viscera, at least hazitaguarana huko ndani. So we don't need friction actually. So such as the digestive tract must be, and then must be smooth. And then the pleural, the pericardium, I hope you know, like pericardium for the cardiac muscles, it has to be smooth. Then we have peritoneum, and if you use Atlas A, you're in good place. You'll see serous membrane how they look. Again, another thing at the most end you need to know is endothelium. So endothelium, actually the circulatory system is lined with a simple squamous epithelium called endothelium, because all those tissues, they require blood. So on the blood vessels, we have endothelium on top of it. So it derived from the mesoderm, and we had already talked about the embryonic germ cell yesterday. So you know when I say mesoderm, what I mean. And also the endothelium will rest on a thin layer of areolar tissue, and then later on, it is often resting in turn on an elastic sheet. So again, I need you to understand that collectively, all these tissue will make up a membrane called the tunica interna of the blood vessels and the endocardium of the heart. So in the heart, we call it endocardium, but the blood vessels, all those tissue which you call the serosa something, that is, they form the intima tunica. So the simple squamous epithelium that lines the pleural and pericardial and peritoneal cavity, we call them mesothelium. So I need you to know, when they align now the peritoneal, the pleural, and pericardial, we refer to them as the mesothelium. When they align the cardiac, we call them endocardium. When they align the blood vessels, we call them intima tunica. So some joints of the skeletal system also, they have the synovial membrane. They are also serosa, and the smooth squamous epithelium. The synovial membranes are also them. So they are made only of a connective tissue, and then the membranes span the gap from one bone to the next, and they secrete slippery synovial fluid that rank them in the same category. And that one brings us to the end. So in our final, very final histological class, which will be very late, and some of you will not be able to get it, that is very final, and then you will quote which topic we need to discuss after that right here, so that I will form the timetable overnight for the next week. So the final class will have tissue growth, development, death, and repair. Tissue growth, development, death, and repair. It will be, I don't know at what time, suggestively, it should be passed. Okay, so first of all, before we finish, because I need to tell something. So before we finish, let me give you this. Before you go, you need to be in position to compare the structure. At home, they are going to be in position to compare the structure of a tight junction and a gap junction, relate their structure different to their functional differences. You also need to distinguish between a simple gland and compound gland and give an example of each. You also should be in position to distinguish between a tubular gland and an acinar gland and give an example of each. You are also supposed to be contrast the merocrine and the holocrine. You need to contrast them and find different method of secretion. You can name the gland that produce or product produced by each of them. And then you also supposed to describe their differences. Yeah, the difference between either mucosa and serous membrane and all other things that we've talked about. Yeah, and all that I don't give, and I will, in fact, if at all we, okay, that is that. So thank you for being here, and we'll see next how the next class will be. So